So don't forget. Okay. It's funny, it's not full screen. Are you? Anyway, I mean, it looks good.
It's
just a
just
funny
that's the
format.
shape that I
Okay.
use. So yeah.
Oh, okay. Well, this is what I
You know,
I do. Yeah, because he did a lot of math bio stuff. He worked for the Akisha, right?
yes,
or
for the long
steady
time.
rate.
Champion looked at it.
Yeah, it was a long time. Yeah. Oh, okay. I don't really get
Yeah,
it.
I don't know. I guess I should change my PDF form, but this used to be the old shape for a lot of projectors.
Right, yeah.
So I haven't really, and it's the PDF letter format. It's just flipped around, so it's easy to print.
Oh,
but,
nice. Okay.
so I haven't, really changed it to
I
fit the new.
no reason to change if it works.
We're not making movies, right? So
Right.
how much width do you need?
I'm gonna put my bag over my teeth.
Oh, and the problem is, why is there?
Yeah. Wait a few minutes for the. They're logging on. It's everything.
Oh. Are you doing closing ceremonies or anything like that?
Let's just do a thank you at the end. Good. I knew there was an optimization seminar. I'm seeing the people coming from the optimization seminar. So.
Yeah, you probably need to go and you might need to go and tell them that some people probably go over time.
Yeah, I think so. We're all right here. We're a little. This room is such a pain. People come in late. It's hard to find a spot to sit. What's that?
Everybody prefers the ends.
Yeah, exactly. And you can't get in. It's a nice size for the number of people. The layout's a bit awkward. Okay.
I forgot to do it.
Neha, could you pull the door shut? Just pull the door shut. You ready? I think we've waited long enough. Well, welcome everyone. Here we are at our closing plenary. And it is my pleasure to introduce the speaker. Alexandar Donev is professor of mathematics at the Courant Institute of the Mathematical Sciences, part of New York University. He did his doctoral work at Princeton, where he obtained a PhD in applied and computational mathematics under the supervision of a chemistry professor, showing an early inclination for interdisciplinary work. He later became Luis Alvarez postdoctoral fellow at Lawrence Berkeley National Labs. He has over 70 publications in Web of Science, garnering over 1,800 citations from other authors, and a recent highly cited paper in Nature Physics. Professor Donev's expertise is in the areas of applied and computational mathematics, computational materials science, and computational physics. He is interested in development of widely applicable mathematical and algorithmic methodologies for the modeling of complex natural systems and applied sciences and engineering. Professor Donev is also an avid outdoorsman, and so I have very much enjoyed our email exchanges about the hiking and camping opportunities in the Okanagan. It is a real pleasure to have Professor Donev here and to have him give the closing plenary address. Thank you.
Thank you, Rebecca. Thank you for inviting me. And yes, yesterday I did get a chance to hike. It's been a long time since I've seen somebody doing handwritten notes, so that's great. Today I'll tell you about some of the recent research in my group in computational biology, but also kind of try to make the talk a little lighter by showing some videos and movies and make it show you a little bit of a broader perspective of kind of the things I've been doing, because it's going to be, I think, out of the expertise of most of you, or sort of because as Rebecca said, I really am sort of a soft condensed matter physicist. And so hopefully it will make sense, but I definitely prefer questions, interruptions if something is not clear, so feel free to ask. I do not need to get to the 46th slide at all. Okay, so the work I'll tell you today, the primary part of the work was done by a fantastic grad student, Ondrej Maxian, and it's sort of computational biology and computational cell biology, I guess. And it's also in collaboration with my colleague, Alex McGillner, who is the biology expert. And then I'll tell you briefly about some work that I did with some colleagues at Lawrence Berkeley National Labs, which is on electrolytes. And I just chose that because it's a current topic, but also there were some electrolyte talks at the conference. So I thought maybe put that in. Okay, so the primary focus of my research for now about 15 years has been fluid dynamics at small scales. So small scales here means nanometer from tens of nanometers. So it should actually be corrected now. We work in tens of nanometers to 10 microns. So small scales. And one of the distinguishing things that happens at small scales is that Brownian motion, which is thermal fluctuations, becomes important and needs to be accounted for. And so we'll talk about that quite a bit. And one of the ways to include Brownian motion in fluid dynamics or thermal fluctuations in fluid dynamics is to use so-called fluctuating hydrodynamics. So that's stochastic PDEs. But there's a lot of mathematical kind of subtleties that I won't go into today. And that's kind of been, fluctuating hydrodynamics has really been the underlying thing that I've more or less worked with for the past 13 years now. And so we developed some fast computational algorithms, but some computational fluid dynamics, but there's also a lot of applied stochastic analysis because, as I said, it's Brownian motion, so there'll be a lot of stochastic differential equations. And so physical systems that we are kind of working on in my group now and in the past a couple of years, and that I'll tell you today very briefly about are suspensions of colloids, so colloidal particles, then slender fibers, so that will be the computational biology part, and also electrolytes. So I'll start with this example, but I won't go show you all the movies. So colloids are basically very small particles, typically micron-sized particles that are in a solvent. So think of many particles, small particles suspended in a solvent. Many materials that you use in your everyday life are colloidal suspensions, such as your toothpaste, for example, mayonnaise is an emulsion, which is closely related. And these are very widely studied in physics because they act as a window into the behavior of atoms. So think of, people like to think of colloids as atoms, but they can be seen under an optical, under a confocal microscope. So we can actually visualize their dynamics and understand many body dynamics in the lab, which we cannot really do with atoms, right? That requires sort of neutron scattering and very complicated interpretation. And so they're very widely used in physics. They also have applications in chemical engineering and so on and so forth. So we studied some colloids recently with some colleagues in the physics department at NYU, where that involved magnetic colloids spun by an externally applied spinning magnetic field. So you have a small particle that's 1 micron size. Think of it as a sphere. It has inside a little magnet, and then you apply a rotating magnetic field which spins that particle very at, let's say, 20 hertz. And so when you spin a particle, and so these particles are also denser than water, so they sediment towards the bottom wall. So they're close to a wall, but they're Brownian, which means they're not that big, so they don't just touch the wall like a heavy part, like a big microscopic particle would. They actually perform Brownian motions, sort of hovering above the wall, let's say a couple micro, on the order of half a micron above the wall. And so now they're being spun and they're doing this Brownian motion. And when you spin a particle above a plane, this is all very small Reynolds number, because these are very small scales. In small Reynolds number, that creates a kind of a vortex, a circular flow field around the particle. And because of the wall, that makes the part, gives asymmetric drag on the particle, which makes it move parallel to the wall. So you're spinning a particle, it looks like it's rolling on the wall, but it isn't. It's actually Stokes flow around it, and the Stokes flow is creating asymmetric drag. And so there were experiments done on this, but I'll just show you a movie because this is just a sort of a preview. So here is, so now I have to disable the spotlight. Sorry, that was the. Okay. It worked, of course, when I tested it just minutes ago. Sorry, I'm just going to start my share again. Okay. So yes, unfortunately, Zoom always does this when I switch to Spotlight. It takes the cursor away, and then I can't go back to it. Okay, so here's a suspension of many of these colloids. Again, they're near a bottom wall. They're being spun by a magnetic field, and this is a simulation that kind of follows their dynamics. And they are performing Brownian motion. It's they're not just moving along a straight line. They're also performing stochastic motion around that. And we studied kind of the dynamics. I won't go into any physical details. I just wanted to show you an example where fluid dynamics and Brownian motion play a role, and where there are experiments and there's some interesting things that you can observe. So a second example I mentioned that are of physical systems of interest to me are electrolytes. So electrolytes, basically your Gatorade, salt water, are just solutions of ions, solutions with ions in them. And so here's, for example, let's say some solvent water in a channel. So there and there's ions, plus and minus ions in the solvent. And the walls here of this channel are charged, so they attract counter ions towards the wall. So for those of you that work in batteries or electrolytes know all about the double layer and the bilayer, so that forms a layer of ions near the wall. And then, for example, you can apply an electric field across the channel to drive current through it. And what that will do is that will, again, push these ions, but it will also create fluid flow because the ions drag the fluid, and it creates a complicated kind of situation where you have some ions carried by the fluid and also by the electric field. And you want to compute things like conductivity through this channel because these are actually used in applications in electrical and mechanical engineering. And so this is called electrohydrodynamics, electrohydrodynamic flow. And so the key kind of in continuum modeling, in kind of standard deterministic modeling, those are modeled by equations called the Poisson-Nernst-Planck equations. And maybe of you may have seen them, may have used those equations. They're a complicated system of nonlinear PDEs. And many of you using batteries may even write papers studying very carefully the asymptotics of these equations and their behavior. But I'm here to tell you that at the scales of the Debye layer, which are very microscopic, those equations don't really work. They're qualitatively correct, but they're not quantitatively correct because they exclude a lot of effects which exist in a real electrolyte, such as there's steric interactions between the ions. The ions don't want to be overlap. When ions come near a wall, they have an image charge on the other side. So there's complicated many-body interactions with image charges. They don't account for fluctuations in the density of the ions. There's no ion pairing, which is counter ions sort of transiently binding to each other. So there's a lot of stuff that happens in real electrolytes that is totally not included in the PDs that you may study, to very extensively, but they will not model the reality. And so what we are kind of working on, and other people, but which I'm working on with collaborators at Lawrence Berkeley labs in the Bay Area, is methods where the water, so the solvent, is treated in a continuum description. In fact, using this, what I call fluctuating hydrodynamics. So you can think of this as Navier-Stokes equations with an added stochastic term on the right-hand side, which accounts for the thermal fluctuations that drive the diffusion of the particles suspended in the solvent. And then, but the ions are modeled actually as particles. So just like the colloids before, which were moving around and performing Brownian motion, now think of the ions being like the colloids. They're little spheres that perform Brownian motion and they move. And so we are tracking all the ions explicitly. They're interacting with each other through steric interactions and other through the walls. But we're modeling we're not modeling all the water molecules, which is super expensive because there's many, many more water molecules than there are ions. And we're trying to kind of see how far you can push this. It certainly is much better than Poisson-Nernst-Planck, more accurate. However, it's not clear. Water is a very special solvent, a polar solvent. So some people believe that for many things, you really have to model the water explicitly. There's a lot of chemistry that happens there. So that's another kind of open question. These are the kind of physical questions we're trying to understand, which is how much of chemistry you need to include in these problems. So I won't. So what are the mathematical challenges to do simulations now where you have ions suspended in a solvent? You need to solve Poisson equations, so to compute electrostatic interactions very rapidly, but with particles potentially in confined geometries like a channel. You also need to solve the Stokes equation. So it's mostly elliptic solves, but with particles, right? So not just the PDs, but there are particles in there. And so that's kind of falls in the category of fast methods, like maybe you have heard of the fast multiple method of Leslie Greengard, my colleague. So that's a method to compute electrostatic interactions very rapidly. So we work with those kinds of methods, but we adapt them to confined geometries. And so one of the key things that kind of we have found is that in order to make this really feasible, you need to be able to reach long time scales. So that means you really need an efficient code. And we have found kind of through students and postdocs that have been very curious about computing, that using GPUs has really enabled us to make these codes really much very, very efficient. And it's all based on FFTs or Chebyshev Fourier transforms when we have channels. But it's all internally done with FFTs, with CUDA libraries, which are very efficient, and that helps us really achieve acceleration. So, for example, we can compute electrostatic interactions between 20,000 charges. So that's 20,000 particles you can think of as charges, and now solving Poisson equation in a channel in less than 6 milliseconds. So so that's a pretty remarkable thing compared to what you can do on the CPUs, even with fast multiple methods. So I just wanted, and the scaling is essentially linear. We have linear scaling algorithms with the number of particles. You can go all the way to 1 million particles in less than 256 milliseconds, which is really not something you can do in existing CPU implementations, even on 64 cores or something like that. So that's just a side note about the importance of computation sometimes, but I will not talk very much about computation. Instead, I'll stop for any questions on this part, and then I'll move on to the main part of my topic, which will be the cell biology aspect. Yeah.
So what's the largest number of parts?
So that's it's a kind of a tricky question, because we can do, sometimes people show that they can scale to hundreds of, like 100 million particles. We can, in principle, we cannot do it on the GPU. You could do it on the CPU. Memory actually is what limits you. You can't fit an FFT grid on the GPU, but that's irrelevant because we want to do Brownian dynamics, which means many, many, many time steps. Brownian motion is a very slow thing. Particles kind of slowly diffuse around. They have to go across the channel and perform complicated dynamics. So at most we can do currently 50,000, 100,000 particles is a stretch, right, to do. So really it's tens of thousands of particles, and that's why these super efficient methods like fast multiple methods aren't particularly useful, because they really reach their optimal complexity when you go to sort of 100,000 or more particles, which is outside of our time scale regime. So, and the GPUs are kind of right at the perfect spot right now to allow us to actually do reasonable time scales with reasonable number of particles. Any other questions? Okay, so now I'll switch to my main topic, which is the dynamics of this cell cytoskeleton. So the cell cytoskeleton is basically the skeleton of the cell. Every animal cell underneath its cell membrane has a structure. You can think of it as a scaffold that keeps it rigid, that allows it to move, to resist, to have mechanical properties. Otherwise, the cells would just squish down if it weren't for that. Plant cells have what's called a turgor pressure, so they're kind of like balloons pumped up by osmotic pressure, but animal cells need this kind of scaffold, mechanical underlying structure, and it's made of very long polymers, biopolymers. We'll call them fibers because they're very long, and there are three kinds of those. There's microtubules, actin filaments, and intermediate filaments. They have different stiffnesses and different lengths and slightly different sizes, and we'll talk mostly about the actin filaments, which are the main component of the cell cytoskeleton. And what's important about actin filaments is that they are almost rigid, but not quite rigid. So you see a typical picture here of an actin filament just in solution in water. It has thermal fluctuations, which means it actually bends just because of thermal energy. So it performs actually stochastic Brownian motion kind of bending, but not that much. It's almost straight. So that kind of made us believe that maybe we can use spectral methods, so things based on Chebyshev polynomials in our case, to describe these kinds of fibers, because they're not super coiled and very bent. You could not do that for intermediate filaments, which are very soft, and they're basically coils like spaghetti, very dense coils. You could not just use a spectral method for that. It's too non-smooth. Microtubules are, on the other hand, almost rigid, and for many applications, you can just treat them as rigid rods. But the actin filaments sit in between mechanically. They're not quite rigid, but they're also not super flexible, and that actually makes them harder for numerical modeling. And so in the cell cytoskeleton, what you have is these actin filaments, which are, again, very long fibers. They have aspect ratio of about 1,000. So they're very, very slender, very, very long. And they're connected to each other by other proteins. They're called cross-linkers. So these are, you can think of short little proteins that just have sticky ends that attach to actin filaments. So you get this thing in the middle where different fibers are cross-linked with each other through these cross linkers. So it's kind of, you can think of it as this is how rubber is also. It's just sulfur connections there between the polymers, but it's basically a network of fibers that are connected to each other with springs. You can think of them sort of as stiff springs. There's also other pieces, which is molecular motors that actually make the cell move and do use energy to do mechanical stuff. We have not yet gotten to including them in our model, but so I won't talk about them. So again, what we have is this network of very slender filaments. So zoom in here. They're very, very slender biopolymers, and they're cross-connected with each other with springs. And now here's a movie. So everything that I'll show you was kind of all the numerics that went into kind of making this movie. Or all the kind of equations also. And so this movie has 700 fibers in a periodic suspension. You cannot see the crosslinkers because there are too many of them. There's many, many little crosslinks. And we're periodically and cyclically shearing this suspension. And what we're measuring is the viscosity, the viscoelastic modulus, the stress that the resistance to shearing this is a very small piece of the cell cytoskeleton, right? We're not trying to model the whole cell. We're trying to understand the viscoelastic properties of a suspension, cross-linked suspension of actin filaments. Okay, so that's what we're going. Hopefully that makes sort of sense. So now, one of the questions that,
Yeah, the 10 scale for Brownian and Brownian.
No, so those are on the order of seconds. What is faster, we believe, is the diffusion of the crosslinkers. So we're not actually explicitly tracking here the diffusion of the crosslinkers. They sort of appear and disappear out of nowhere and bind with the idea that they had diffused rapidly through the system. That's also perhaps questionable. And then I have another project, but that's done by undergrads and master students. So it has a very slow time scale of progress on which, where we actually do explicitly model the diffusion of the crosslinkers. But the unbinding scale is 1 second. So it's not, which is kind of relevant for the biological systems. So it's not very fast. Any other questions? Okay. So one of the kind of, we wanted to answer some physical questions to understand things. And one of the things that we notice is that the current models used sort of in the biology and computational biology world are very simple. They kind of make chains out of little rigid cylinders connected to each other with kind of spring hinges, and they never include hydrodynamics. They sort of have like some sort of drag on these particles, but they never actually include full hydrodynamics. And so, but there are very well-known examples in biology where you can get macroscopic flow out of, even though for example, in the cell, when the cell divides, you can just look at it under the microscope, you see very large scale on the cell scale, so that's 10 micron scale. And we know that these are created by kind of motors plus these actin filaments. So we know that kind of these fibers in a suspension can create large scale flows. So that's what we call hydrodynamic interactions or hydrodynamics, long range hydrodynamics. And we wanted to know whether these long-range hydrodynamics interactions play a role in the rheology, so in the viscoelastic behavior of these cross-linked actin gels. So that's kind of the physical question we're going to try to answer. And if I get there, I'll maybe give you some answer to that. It's a very difficult problem because it's very multi-scale, very, very slender fibers, many of them. So if you try to resolve all the scales, you will quickly run into issues. And again, the very hard thing, which we're still working on, is that these fibers actually also perform Brownian motion, as I mentioned. So they're actually kind of not just, they kind of are fluctuating, you can think of them. That is not included in what I'll tell you right now. Maybe at the very end, I'll tell you why that is extremely challenging. Hopefully in a year, we'll have something to say about it. Okay. So in the interest of time, here. I will skip the Brownian dynamics part, which was for the ions and the colloids, which I showed you in the beginning. You can ask me later if you're interested in sort of the equations of Brownian dynamics. And instead, I'll just switch to modeling fibers, networks of fibers in Stokes flow. So I'm sort of, so if you have a fiber, a very slender fiber, like an actin filament, again, it has aspect ratio 1000, and you want to model it, and it's in a solvent, in a fluid. The simplest way, which is what almost always is done in sort of the physics, the soft condensed matter physics literature and polymer physics, is to just represent one of those fibers by a chain of beads. Yeah. Which is the leftmost picture in the screen. And so, but the problem with that is that these fibers are very, very slender. So you need 1,000 beads per fiber to do this. And it's very expensive. And typically what people do for flexible polymers like DNA, they do what it's called coarse graining in physics. So they take a big chunk of DNA and represent it as one bead, right? So it's really a coarse grain model of the DNA. They're not actually resolving the whole backbone, which would be super long. But for actin, you cannot do that. You cannot represent a very slender, almost rigid filament. By this coarse graining is it gives you the wrong hydrodynamics. So again, physicists would like to go in the opposite direction, which is to coarse grain this, to have fewer degrees of freedom per fiber. But we asked a kind of a different question, which is, what if I now add more blobs instead of less blobs? So what if I make a fiber out of these? I call them blobs or beads. And what if I add more of them? And in fact, what if I go to a continuum limit where I have infinitely many of these blobs along a curve, the advantage of that is that once we have a continuum description of the fiber, not a discrete one, then we can use our tools like Chebyshev polynomials, like spectral methods from PDE, numerical PDEs, to represent this fiber now with 16 degrees of freedom. So 16 Chebyshev nodes, which are these big yellow things. So again, those are not physical degrees of freedom. It's not like coarse graining a part of the molecule. They're just Chebyshev nodes along the fiber. So that was kind of the idea that we had. Okay, so so now we need a mathematical model. So now I'll start writing equations. It is a math conference after all. And so all of these slides will have two equations. One will be the discrete equations, which is again for this model where you make a chain out of discrete beads. And another equation will be the continuum limit of it. And so even though I'll show both on the slides for reference, just look at the red equations, which is just the continuum equation, so that we don't try to go through both of them. So the first thing that we want is the fibers to be inextensible. So what does that mean? So we're going to describe a fiber by a function x of s. So that's the position of the center line as a function of the arc length s, which goes from 0 to L, the length of the fiber. And the tangent vector is just the derivative, dx ds, and the inextensible means that the tangent vector always has unit length 1, which means the arc length parameterization is preserved by the motion of the fiber. You cannot stretch the fiber, and this is a thing that actually is relevant to biological fibers. They're very hard to extend, but much easier to bend than to extend. Biological fibers also twist. We have also recent papers where we include for the twist. It's unclear. So biologically, that's important because fibers are chiral, and so they actually that might be connected to the emergence of chirality in cells. Here, I will not include the twist of the fibers. It will just be represented by a curve. Yeah, and so the first thing you need is elasticity. So to model the elasticity of the fiber, what you have an energy, which is the elastic energy of the fiber. So it's a functional, and it's just the way it's modeled typically is as the integral of the curvature squared. So this is called bending energy, and it's a typical way to model an elastic, like a beam in civil engineering, a bead in a bridge, for example, right? A beam would be modeled like that. So it's the energy is the integral of the curvature squared. And that means when you take the functional derivative of that, you get the bending force, which is the fourth derivative of x. So the bending force density on the fiber is XSSS, and this is what tries to make the fiber straight. So you immediately see it's very stiff. It's a fourth derivative. If you try to bend the fiber a lot, it will very quickly, it will very much resist that. And there's some boundary conditions which depend on the circumstances. Now, as I said before, the fibers are inextensible. That means internally they have some tension. If when you put them in fluid flow, the flow may try to stretch them, but they will resist that. So they'll have an internal force density lambda, which will be a Lagrange multiplier for inextensibility, and that has a form tension. So T is a scalar function, T of S is a tension times the tangent vector and the S derivative of that. So again, that's just mechanics looking at force balance between two pieces pulling a little segment of the fiber. So the two forces that act on the fiber are bending force and stretching, resistance to stretching, which is this Lagrange multiplier, lambda. So now that's the mechanics of the fiber. Now we need the fluid dynamics, because now the fiber is in a Stokes fluid. It's immersed in a fluid, so we need equations for the fluid. And so again, I will write. We should just look at the red equations here. So the approach, these equations come. I mean, there's a very long history, but let's just say that for me, they are motivated through the immersed boundary method, which is something that my colleague Charlie Peskin created in the 70s to model heart flow. So it's a standard way to model soft bodies immersed in that interact with the fluid or immersed in a fluid. And so for those of you that know the immersed boundary method, this will look like the immersed boundary method. So what are the equations? So we're going to have a Stokes equation for the fluid flow. So that's the usual steady Stokes again. We're going to be in the zero Reynolds number regime. So gradient of pressure equals Laplacian of the viscous term. And then we're going to take this force density, which acts on the fiber, and we're going to spread it to the fluid by using a regularized delta function. So the idea is that the fiber feels a force, but that force is just spread to the fluid. There's no real distinction between the fiber and the fluid. They're really one structure. So you can think of this as fluid everywhere model, and you're not actually resolving the structure itself. It kind of looks strange at first. It seems like it has to be wrong, but I'll try to show you later why it's reasonable. So again, we have an integral here from 0 to L of this force density in the fiber, which just goes on the right-hand side of the Stokes equation. Then you solve that equation and you get a velocity of the fluid. So you get a V, the velocity of the fluid. Then you need to move the fiber, and the fiber simply is advected by the velocity. So it just follows the velocity. So to compute the velocity of the fiber, which is the time derivative of the position, I take the velocity field and I convolve it. I locally average it with the same regularized delta function that I used to spread the force. That's actually key, that those two are the same. It gives you energy conservation. It makes these equations have the correct physical structure. And again, the force density F is just the sum of bending and stretching forces. So now we have, these are the equations that we're going to try to solve. Okay, you're given X, the fiber shape, that gives you force density. You need to put in the Stokes equation, solve it, and move the fiber. The complication will be that the fiber is inextensible. So this is a Lagrange multiplier, which we'll need to solve for. It is not something that is given. It is given to us. Okay, so any questions before I go on? Oh, it's chat. I had it open here and then I closed Zoom. So. Nope. Okay. Good. Okay, so how are we going to solve these equations, or how are we going to proceed? So the first thing is, these are elliptic equations, right? The fluid equation is elliptic, which means there is a solution operator for it, and we can just use that to eliminate the fluid velocity and write an equation for just the fiber, which takes into account the fact that there is fluid, but we are going to kind of try to avoid the fluid. And in particular, if these fibers are in kind of unbounded fluid in an unbounded domain, then we just know the Green's function for Stokes flow. We can just use that to solve the fluid equation instead of actually solving it on a grid or using some solver. And so I'm going to introduce this kernel, this function, which is basically the Green's function for Stokes flow, G, regularized from the left and the right by the delta function. So this is what this function tells you is if I have one, we call them blobs, so one regularized delta function, I apply a force on it that creates a flow, and then I have another one of these blobs. What's the velocity of that other blob, right? So it gives you the ratio between force on one blob and the velocity on another blob. And it's just the Green's function, but regularized, so it's not singular. It's singular anymore. And I forgot to say that the regularized delta function, the support, the width of that function, you can think of it as a Gaussian, is related to the thickness of the fiber. So it's a physical, it's not a numerical regularization like in the classical immersed boundary method of Charlie Peskin, but it's actually physical. This is the thickness of the fiber. The fiber is not a line, it has a finite thickness. And so, so now I'm going to define an operator, so which we call the mobility operator. So that takes a force density and gives me velocity. So remember, I know the force acting on the fiber, elasticity, stretching, but I want to find how it moves. So I want to find velocity. So this operator maps between force densities and velocities, and it simply just takes a force density and integrates it against this kernel. Okay, this regularized Green's function for Stokes flow, and it's an integral along the length of the fiber. So now this is again a non-local operator. So it's an elliptic kind of operator, but it's a non-local one. And so now we're ready to finally write an actual equation for the fiber, just a single fiber right now immersed in a Stokes flow. So here's the equation. Xt is force density, which again, remember, is depends on, it's the fourth derivative of x, acted on by this non-local hydrodynamic operator. Okay, and lambda is a Lagrange multiplier, which has to be chosen to satisfy this inextensibility condition. So this tau, remember, is x sub s. It's the arc length derivative has to have constant length. So it's a non-local elliptic PDE with a constraint. Okay, so not a trivial, not necessarily trivial, but it's one-dimensional at least, right? So that's that makes it considerably. So I think like makes it possible to solve this kind of equation. And then you could sort of ask questions: is this PDE well posed? And actually, even that question is not really answered kind of to the satisfaction of analysts, but we kind of numerically believe that yes, that the fluid velocity converges pointwise strongly up to the endpoints of the fiber, but lambda itself, the Lagrange multiplier, only converges weakly. So only moments of lambda converge, but we believe that this equation makes sense. And that we can actually solve it. Okay, so and then I'm gonna tell you a little bit about the numerical methods we use to solve this equation. Okay, so remember that this kernel, so this operator, involves an integral, a convolution of the force density with this kernel, and that kernel was the regularized Green's function for Stokes flow. And it turns out that for some special choice of this regularized delta function, you can analytically compute this. So you can think of this as just being something given to you in an explicit analytical form, but it is long range. The Green's function for Stokes, just like the Green's function for Poisson, decays like 1 over distance in 3D. That means fibers interact with other fibers hydrodynamically through the fluid flow over long distances. That's why hydrodynamics is long range and difficult, just like electrostatics. So this kernel is something explicit that decays like 1 over distance. Okay, I won't write. There's an explicit formula, but you don't need to look at that formula. And so now, what I told you so far kind of seems like just a model. Like we kind of came up with some model. We said this is our fiber, but then you can ask, well, how does this connect to mathematics to, more, let's say, asymptotics to more precise calculations. So in the field of in this field, what's used to model very slender fibers in Stokes' law is slender body theory, which is an asymptotic theory for very slender bodies immersed in Stokes' flow. And it has a long history, primarily because of things like flagella, again, applications in computational biology. And so, the way you do that is much more complicated. You consider, let's say, the fiber to be a cylinder with boundary conditions, so no slip boundary conditions on the cylinder, and it's a very slender thing. So you do an inner expansion and outer expansion, you match them. All of the asymptotics people know what to do. But it's quite a complicated thing because it's a curved fiber, and you need to do all the expansions around there. But it exists. It's been known for a very long time, and so there's some formulas. And so, but we approached it differently. We said, what if we take the integral I just wrote, which was our model, and we do asymptotics on this integral? So now what we're going to assume is basically that the F is smooth, right? It varies slowly along the fiber on a scale much longer than the thickness of the fiber on this regularization. And we're going to do asymptotics on this integral, and the same thing, inner outer expansion, but now it's an expansion in S, not outer and inner expansion in 3D space, but it's just in S. And you do that, and I won't go through the details, but what you get, the formula you get from that, is exactly what people call slender body theory. So that's a simplified formula that goes from force density to velocity. So it's a kind of an asymptotic expansion of that integral above. And it's, again, known in the literature since bachelor, so like the 80s, but lots of people then improved it in many ways. Okay, so and so the reason I'm just showing this is to say that even though we wrote a model that seemed like it was just made up, it does go to the standard result, but you have to choose the radius of the blobs, right? The delta function that we used, you have to choose its radius carefully to how it's related to the actual radius of the fiber. Okay, and magic number, turns out it's 1.12 times the size of the fiber. So the blob radius is slightly bigger than the cylinder, And the important thing is that our approach, which is just writing this integral, unlike slender body theory, immediately works for multiple fibers, because we just use the same thing, but now it's an integral on the other fiber, but you're evaluating on a point on another fiber, right? So the target, this point, is on another fiber, but the integral is over another fiber. So this just immediately works. It also actually fixes many issues in slender body theories, because the operator that appears in slender body theory is kind of ill-defined at the endpoints of the fiber. It also is actually not symmetric positive semi-definite, which means that it's not dissipative. So now you have it's not really an elliptic equation. So that leads to instabilities. And so, but this thing that I wrote is actually guaranteed to be symmetric positive semi-definite. And it fixes all kind of a lot of issues that there exist in slender body theory. Okay, any questions so far? Yeah. Yeah.
Your special blob radius, is that like it turns out to be that? As in, sorry, is it that you have to choose that number to make them agree, or is there some like fundamentally apply?
So it depends on the delta function you choose, right? So remember, we represented, instead of a delta function, a regularized delta function. So depending on which regularized delta function you choose, the width of that will be proportional to the radius of the fiber, but the constant will differ depending on your choice. This is for the specific choice of delta function we have taken. It's this relationship. And the way you think about it is what you're trying to match is the total, like you take a cylinder and you just drag it through the fluid perpendicular to it, and you measure the drag on it. And you want to match that to the actual drag on a real cylinder in Stokes flow. So that's really, it's the inner expansion that you need to match, which is the inner expansion is the flow around the cylinder, because locally you assume the fiber is straight. Yeah. Okay, so now, so that would sort of nice, except we're missing two pieces. So the first piece we're missing is I wrote that integral above, And I told you it's better than this slender body theory that other people have written, but I didn't tell you how to evaluate that integral efficiently. And the reason that's not easy to do is because this kernel, it's a regularized Green's function. So it's not singular, but it's almost singular. The length scale in that kernel is the thickness of the fiber. And so you have you're performing an integral over something of length 1000 with a kernel whose width is 1. So that's still a challenging integral to evaluate because it still has a hidden length scale, small length scale in it. So that's part one. I'll try to show you that as well, see how I do with time. So we have some ways to do that. That actually comes from the world of fast methods of sort of boundary integral equations for Stokes flow, where they're have very smart ways of doing quadrature, because there you have to integrate actually singular kernels. There they actually integrate the Green's function for Stokes flow on a surface, so that's a singular kernel. So they have this smart singularity subtraction techniques, and we've adapted them to kind of this setting. That's one piece we're missing. The other piece we're missing is the inextensibility constraint. How are we going to handle this constraint to find this Lagrange multiplier that we need to enforce inextensibility? And so that part is was the main contribution in the beginning, like the first paper that we wrote. So I'll try to explain that. And yeah, feel free to ask me any questions. But now I'll actually go back to discrete fiber because this is easier to explain with the discrete fiber first, and then I'll write the continuum equivalent. So imagine we have a bunch of points, which are these blobs, connected with rigid, inextensible segments, right? So they can move, but you want to keep the length between them fixed. So how will you do that? Well, it's clear that if I want the distance between these two blobs to remain fixed, the only thing I can do is I can rotate the vector connecting them, right? The only motion that preserves inextensibility is motions that just rotate the links. So that means that the difference between velocity of the two blobs, so the relative velocity, has to be some angular velocity cross product with the tangent vector. So again, only inextensible motions are rotations. So the only motions that preserve a vector having unit length being on the unit sphere are rotations on that unit sphere. So now we go to the continuum equivalent. So imagine that I'm given a angular velocity along the fiber. So every point of the fiber is going to rotate with some angular velocity. And I cross that with the tangent vector. And then I have to add up, because this is every little segment, right? Every little piece of the fiber gets rotated. So if I start from the beginning, I have to integrate to the point where I am, because all the rotations add up. So to get the velocity of the fiber at a point S, I need to integrate again the velocities that come from rotating the fiber. So what the statement here is that any inextensible sufficiently smooth motion of the fiber can be represented in this way. So this is a parameterization of the manifold of inextensible curves. Okay, so it's an explicit parameterization. It says the only way to update this curve is to rotate the tangent vectors. And I call this right-hand side is an operator. It takes an angular velocity, integrates it, it gives you another velocity, linear velocity. So I call that operator script K. So this is called the kinematic operator. It tells you how to go from it parameterizes again the all allowed motions of an inextensible fiber. And so then there is another principle from physics, which it's basically just in calculus of variations, it's just a first order optimality condition. But the way physicists would say it is that the principle of virtual work, which the way physicists would say it is that the constraint forces cannot do work. So again, this is an inextensible fibers. We're trying to stretch it, and it will resist that, but it cannot do work. There's nothing consuming energy in the fiber to resist the motion. So you cannot get energy out of this Lagrange multiplier. So that means for any allowed motion of the fiber, the constraint forces cannot do work. And that turns out mathematically to be equivalent to saying that the adjoint of this operator K, the L2 adjoint of the operator K, acting on lambda must be 0. So again, that just means that the constraint forces are, you can think of them as sort of tangent, perpendicular to the manifold, and they don't do work when you move along the manifold. So now we have all the pieces to write now equations that we're actually going to solve on the computer. So I'll go through this again. So we have the bending forces and the unknown Lagrange multipliers. They get acted on by this non-local hydrodynamic operator, right? Non-local elliptic operator. And that must be, that's the velocity of the fiber, but that must be an inextensible motion. That must be written as K times omega, where K is the kinematic operator. And at the same time, K star lambda, the adjoint operator of K, must be 0. So these forces don't do work. And so that's this red system, it's a saddle point linear system, the kind of typical thing you encounter in optimization. When you solve that, you solve for omega, which gives you the motion of the fiber, and for lambda, which gives you the tension in the fiber that is resisting your motion. So at every time step, you need to solve this non-local PD, but we have converted it now to a linear problem. Okay. And so what's the reason for this approach? So the reason we took it is because it strictly preserves the inextensibility of the fibers. So our fibers never become never stretch, because if you allow that, then typically people have to put a penalty force to kind of bring them back. That introduces stiffness, and then you go into another bag of worms. And so we decided not to do that, but instead to keep the fiber strictly inextensible by only rotating tangent vectors. So instead of moving the fiber, we're rotating tangent vectors, which strictly preserves their inextensibility. It also makes the linear algebra, exposes the saddle point, symmetric saddle point structure, which it preserves, there's a structure to these equations that it preserves. Okay, so those are the equations. And now we need to solve them to and then update the fibers. So there's, again, a lot of steps in here. So I will skip kind of a lot of the numerics, and I think I'll show you just the asymptotic evaluation of one of those integrals. So we use spectral methods, Chebyshev-based representations for this equation. And it's a collocation method at present. Now we have we're working on Galerkin. We're finding that those are better, but kind of reasonably standard. If you know spectral methods for PDEs, it's reasonably standard. Again, the challenging things are this is a non-local operator that you have to be able to apply efficiently. So that's a quadrature problem. And I'll come back to that in a second. And then every time step, you need to solve linear systems, which are dense because, again, this operator M is non-local. It couples every fiber to every other fiber, right? So you need a preconditioner, and then you do typical games in the preconditioner. You neglect some of the far field interactions. You only include one fiber interacting with itself. So you need, so there's some linear algebra that you need with this, and a quadrature. And then there's time stepping. That's standard, something like backward Euler, BDF2 kind of, you need to use an implicit scheme because it's very stiff for the bending. But we also do some splitting where we split some of the hydrodynamics into an explicit part and only the local hydrodynamics in the implicit part. So typical IMAX type schemes so that your linear solves are cheaper. So again, but all really standard methodology and kind of spectral method, numerical analysis. I mean, you need 2 courses in numerical analysis to follow all of it, but it's relatively standard. So I won't go through that in detail right now. Okay, so now instead, I guess I didn't put the quadratures in here, so I'm not going to tell you that. Let me show you some just a very quick physical results. So again, reminder, now we have this numerical method. Again, not working, but I showed you this movie earlier, so I guess I don't have to stop it. So we have a network of many fibers. Here it's like 700, I think. It's a periodic system. So because we're only modeling a little piece of the actin cell, they're connected with little springs, which you can just add as an extra force acting on the fiber. So it just goes in the right-hand side. Those springs are treated explicitly in this numerical method. They can be done implicitly, but the code would have become just too complicated for a grad student, so we didn't do that. Okay, so just to zoom in, I don't know if I can play this video. I don't know. It's not playing. Okay, so just to zoom in, this is only one fiber, the red fiber, and other fibers around it, and all the crosslinks are these black lines. So again, we're cyclically shearing it. It's moving, but very little. The springs are being stretched, but very little. So we're just sort of shearing this actin gel. And what we're measuring is the total force, the total stress. And so when you do that, what you can compute from that is just by shearing at different frequencies, you get what's called the viscoelastic moduli. As a function of the frequency. And that's kind of this thing that we were after. That's what you need in order to write a continuum model for now a big system, kind of if you want to model the whole cell and you want to include the explicit to the cell cytoskeleton, you would need these. But what we found, the important thing I want to emphasize, not the physics of this, is that we did simulations where we included and we did not include the hydrodynamic interaction. So we include one simulation where fibers only interacted with themselves through the fluid, but not with other fibers, and others where it interacted with everything. And we found that when we took the ratio of those moduli, G without hydro, with hydro, we could get a difference of about 25% sometimes, so for some frequencies. So that means that these non-local hydrodynamic interactions, while they're not like, they don't contribute the majority of the viscoelastic response, they contribute a non-trivial fraction of the viscoelastic response in this actin gel. And then here's just another movie. Hopefully this one will play, we'll see. Oh, I had to click double. Okay, I guess that was my mistake. Okay, so here's now another movie where it's dynamically cross-linked. So now what you sort of, these black little lines, which are the small cross-linkers, the little springs, dynamically, randomly attach and detach as we're shearing this gel. So there's another component of dynamics here, which is dynamic cross-linking. When you do that, what you find, and this is actually observed in experiments, is that the fibers tend to bundle together. So once two fibers cross-link, the cross-linkers pull them together, and that makes other cross-linkers more likely to bind, and then you get bundles of fibers, which you actually see in a microscope. And so these are now bundled fibers. And now what happens is in these bundles, the fibers are closer to other fibers. And so they interact hydrodynamically through the fluid stronger inside a bundle than they did before. And so in this case, when we measure the viscoelastic moduli, now we find 50% sometimes difference in the moduli with full hydrodynamics and without full hydrodynamics. And we're pretty sure that that comes because of these bundles where other fibers are pretty close to other fibers, and one fiber moves, it drags the fluid, which drags the nearby, entrains the nearby fibers in the fluid flow. Okay, so I'll stop here. So just to emphasize here that adding thermal fluctuations is extremely challenging because for several reasons. Let me just give you two. The first reason is kind of, let's say, numerical. The easy reason is that fibers are no longer smooth. They're actually, the spectrum, if you look at Chebyshev space, is supposed to decay like a power law, 1 over wave numbers to the fourth. That comes from, because of the bending forces, the fourth derivative. So those fibers are not smooth. So it's no longer clear that spectral methods are actually useful to solve that, solve those problems, or if they can in fact work. So there's numerical issues. Then there's actually mathematical issues, which are much deeper, which is that we don't actually know how to write an equation, a stochastic now PDE, that actually makes mathematical sense and is modeling a fiber that is fluctuating. So we neither have equations, nor do we have numerical methods to solve them. So you would say we're sort of doomed, right? But we like a challenge, and this grad student is great, and he already published enough papers to get a PhD thesis, so he's kind of willing to spend his last year now with this problem. What you can always do, so you can ask, well, since you don't have an equation, you don't know how to solve it, what are you going to compare against? Like, what's your reference answer? And so the idea then is that we're going to go back to what the physicists trust, which is the bead model, where you do make a fiber, but now we're only going to study one fiber fluctuating. Now I can't do 700 fibers, but we have methods that can simulate the Brownian motion of an inextensible fiber made of beads. And then we're going to study the statistics and then compare to what we get from our spectral methods, if they ever work, and hopefully make progress that way. So that's sort of the next year challenge for us. Okay, so that's it. Thank you. And here's some references if you want to.
I have one that I probably should have asked way back at the beginning. Why does having a fourth derivative mean that it's more resistant to bending?
It just means that high frequencies get damped very quickly. So if you want to wiggle it a lot, it resists. That's why the spectrum is 1/wave number to the fourth, right? So it decays pretty rapidly, but it's still a power law. But it doesn't like to be very curvy, right? It likes to kind of bend at the small wave numbers at the large scales.
And that's part of what makes it stiff as well.
It's also stiff, right? Because those modes have a super fast relaxation time. So you have to use an implicit method. And then when you do fluctuations, that's a whole big problem in itself, because typical implicit methods like backward Euler damp that, but that means they kill fluctuations also. And so it's this problem of everything you want to do in numerics to kill instabilities, you don't want to do when you have fluctuations, because they kill what's called fluctuation dissipation balance. So there's a lot of, a lot of problems. So we'll see if we can solve some of them, but it's pretty challenging. Mm-hmm.
Have you thought about modeling other types of hydrogel like that in mucus specifically? And then I guess I have a follow-up to that question.
I mean, so the methodology we have developed is sort of suitable, not just for actin. In fact, actin, while we always use it as the motivation and you put it in grants, it's an extremely complicated thing. Actin is a polyelectrolyte, has a lot of chemistry. The fibers actually grow and shrink, they polymerize, they depolymerize. And there's no two experimental measurements of the viscoelastic behavior of an actin gel, even in vitro, right, with reconstituted, that agree by even an order of magnitude, right? So they agree by factor, like sometimes 2 orders of magnitude, right? You measure one and that's because actin is a super complicated thing that tends to age, has chemistry, like you have to really stabilize it very carefully. And anyway, so in fact, other materials might be much simpler, where you can actually compare to experiments, as long as they have kind of these fibers. But one thing I didn't mention is that the approach I presented explicitly assumed a linear chain. So even if I tried to close the chain to make it circular, that parameterization of inextensible motions breaks down, because then I have to come back where I was. So even if I rotate tangent vectors, that's not enough. There's an additional constraint. So branched polymers and other more complicated things are not within the scope of this method. It was meant for actin, meaning really stiff, almost straight linear polymers. So we have used some specific things, but some of the stuff we've developed could probably be generalized to other things. But we're really, most of Alex McGillner, my colleague, is interested in actin, and I go along with what they're interested in.
So why are you so convinced about the time scale of cross
The diffusion or the?
I just know the binding and unbinding time scale of those crossing and mucus, it just expands like many.
I'm not convinced. So again, it's going to be a cop-out answer, but the parameters that we used here for the binding and unbinding, you can say they come from experiment. However, it is extremely unclear what the experiment actually measures. And the only thing they can measure is unbinding. And even that's questionable, but they claim that the unbinding frequency is on the order of 1/2. This is like what in kind of biology literature and experiments. The problem is very challenging because it can unbind but then rebind again. So what exactly they're measuring is very unclear. And we made the binding rate simply be such that at steady state, you get a number of cross-linkers per fiber that seemed physiologically relevant. Okay, it is not from my first principles what the binding rates are. I think that's a whole other problem in reaction diffusion, which is these are diffusion limited reactions, so they're quite complicated, and you need to kind of understand the rates of binding of some cross-linker diffusing through this complicated gel and trying to bind and unbind. And I think that's another challenging
You
issue.
can fix the equilibrium fraction and change the time scale. Binding in the
Yeah, that's what we did. We fixed the equilibrium fraction, but we just put one unbinding scale, which was this 1/2 seconds, which is or 2 seconds inverse, which is.
If you haven't added the Brownian fluctuations, but you do have random from the random.
Yeah, I mean, I didn't go through that. We have added the fluctuations for rigid fibers. So in that movie that I showed you, this one, these are actually rigid fibers, and they are actually performing Brownian motion. I remember I told rigid is much easier than bending because rigid has only 6 degrees of freedom, and that's well understood how to do Brownian motion of rigid fibers. So these were actually rigid, and then we do have full Brownian motion, but we don't have bending fluctuations yet. So that's the challenge.
Okay, any other questions from Zoom land or the room?
Okay, thank you guys for listening,
Okay,
staying until the bitter end.
Speakers. And thank you all for coming to an in-person conference. Good to be back.
Thank you for organizing.
You're welcome. It was
It's been great, yeah.
See you in Fredericton in 2023. Don't forget your lanyards. Please leave them in the bowl at the door on your way. That was fabulous. Thank you. And you can stop the recording here.
I want to do a PhD in your group.
Yeah.